Okay, so what do we get? Make sure I don't run out of time. So basically want to show you some of the things we were able to see in this. What's interesting is it's not just the genes that tell you what's going on in the system. You can see the genes as a whole, and it kind of gives you an idea of what's going on as well. So in this instance, in this graph, we've got our different viral strains from least severe to most severe. We also have day post-infection here on the x-axis. And what we're doing is I'm looking at the number of genes that were significantly different at each of these time points for young, adult, and aged. And what you find is the stronger the virus, the more the genes change, right? Here we have this positive negative control, doesn't really affect the cells. We're not getting much change in gene expression compared to controls, but as we get more severe, you can tell the number of genes go up. Not only that, but it starts going up earlier in at least the adult and age, which brings me to as you get more severe viruses, you see increased gene expression. But what's interesting, especially at this last time point or this most severe form of the virus, you can see at day one, the young are basically not even, hardly inducing any genes. But here in the adult and the aged animals, there's quite a bit of induction. So in a way, this would kind of suggest that, these younger animals, don't really recognize the virus right away, but in the Asian adult, definitely are seeing that, are responding to the virus much earlier than the young. We can do some unsupervised machine learning. I was looking at some of the talks. I didn't see a whole lot of PCA plots, but I love PCA plots, principal component analysis. What you can do is you can basically, on all those biosets, those 74 different groups, we can take those comparisons, look at the gene expression, and group them in three-dimensional space. We kind of summarize that transcriptome in three-dimensional space, three ways. And so that genes that are or transcriptomes that are most like each other are closer together. Those that are more distant or more dislike each other are farther apart. What you can absolutely see here, this is days. We did was color it days post-infection. You can see that this is by far the variable that's having the most effect on our data. And what you're also seeing is that, you kind of got two different phases of the virus. You have the early phase here, which encompasses up 2 days post-infection. And then you have this 4 days and 7 days after post-infection, kind of a later stage. This might be the virus reproducing, and this might be clearance here. Because what happens is by 7 hours, it's starting to look more like the control animals. And if we basically look at some of our variables and the effect, the F value, the higher the F value, the more effect that variable has on our data. You can see DPI is much, much higher than everything else. But what we're also seeing is we'll see an effect. So error would just be an F value of 1. The only other variables that are higher than our error would be age of the animal, which would affect the expression, and the type of variant of virus used. What is nice is that this GOID represents the different studies we used, and it really doesn't have an effect. So what this tells us is our normalization probably worked well because we're not really seeing effect between different studies in there. Again, I mentioned, we've got two different things. We've got a kind of early phase and a late phase. Person in our group was really good at machine learning, which is really hot now. He did a binary elastic net analysis on our stuff with a bootstrap. What he found was you could basically characterize these into early stage genes and late stage genes. And when you actually graph all of these out, you can see there's a lot of connections. This was Pathway Studios. They're no longer around anymore, but it's a way to basically use their database to find connections between inputted genes. You can see that the early ones tend to be on the outside of the cell, which I thought was very interesting. But what I want to do is I want to spend some time with this one, this gene ADM, aden, I can never say it. It's on my next slide, so I'll pronounce it then. But this ADM gene, we were very interested in. Again, it was a very early stage gene. We thought it might be important. So we started looking up some things on ADM. What is nice is that as a bioinformatician and somebody who loves to use other people's data, other studies were coming out as we were doing our work. There was a particular study out there looking at proteins in blood of COVID-19 patients, severe COVID-19 patients. So what we did, we downloaded their data and just basically said, okay, what is this adrenomedullin? What does this gene do, in their study? Again, we're trying to take our mouse data and see what it's doing. You relate that to the human condition. Here you can see these are days on the x-axis, days after admittance. And what you'll see with this ADM is that in blue, these are patients that died within the 20 days after being admitted. Blue would be intubated, ventilated, so not as severe as death, but pretty bad. Green, they required oxygen, and then the blue one didn't require oxygen. Oops, sorry. And then the orange, they weren't hospitalized. As you can see, just that when they're admitted, it almost, it's ranked based on adrenomedullin expression totally relates to the severity of the disease. And not only that, but as you look at, there's absolutely an age effect. So if these are different age groups going up in age, and you can see that even at basal levels of expression, they just keep going up and up as you get older. So maybe, adrenomedullin, maybe that's somehow, associated with the age effect we see. I just thought that was interesting. And later on, a lot of people published on this. We weren't the only ones to see this. Again, we saw this in mouse, but people were seeing this in humans, showing that it has absolutely increased with severe COVID-19, and that, you might be able to use this as a predictor of mortality. If adrenomedullin levels get really high and say your patient, you need to pay close attention to them because they have a good potential to die. And just before, talking about the different things that we can see, I just want to mention that we have, if you go to our paper, we have an interactive portal. It's at this link. You can actually go in and visualize the data yourself. We thought, I've seen so many papers out there, you have to download and do all kinds of stuff to the data. We make it easy for you. You can actually just go to this site and you can look at PCA plots, you can do line plots, all kinds of different things that you can actually take the data that we've collected and analyze yourself.